Disorders of the mind have long remained mysterious and largely neglected. One of the most storied and controversial of these disorders is schizophrenia, a psychiatric disorder involving impairments in cognition, perception, and emotional well-being that tends to manifest late in adolescence. It afflicts approximately 2.6 million, or 1.1% of all adults in the United States aged 18 or older. This is more than the population of New Mexico. These individuals are often disempowered and pushed to the edges of society. For example, the NIH estimates that 40% of affected individuals will never receive treatment for their condition. Despite having a clear pattern of heritability, schizophrenia has eluded attempts at identifying genetic variants related to increased risk for decades, causing it to be known as biology's black box. However, 2 years ago, a global team of scientists known as the Psychiatric Genomics Consortium collected genotypes at 9.5 million loci from over 36,000 schizophrenia patients and 110,000 controls, the largest such study to date. They combined this information with available ancestry information and used a statistical model to look for relationships between particular genetic variants and the development of schizophrenia. The study found an impressive 108 risk variants, many of which are quite common. Each of these variants individually have low effect on risk, but seem to collectively contribute to the development of most cases of schizophrenia. By far, the most strongly associated of these variants was found in multiple SNPs across the region of the P arm of chromosome 6, known as the Major Histocompatibility Complex, or MHC. This is an area known to contain more than 200 genes central to the acquired immune system. While this diffuse association with the MHC did not map to a single disease locus, some regional cohorts displayed a series of SNPs within the C4 locus that had a particularly strong association with the development of schizophrenia. As such, the McCarroll lab at Harvard Medical School decided to consider the complex structural variation at the C4 locus more closely. C4, or complement component 4, is a protein involved in innate immunity, tagging pathogens for phagocytosis. It has 4 different common structural haplotypes, stemming from 2 different alleles, C4a and C4b, which can come in either a short or a long form containing an intronic insertion of an 11 kilobase transposon. The McCarroll lab analyzed the C4 locus in 222 chromosomes obtained from the HapMap project to give precise measurements of C4 allele type and copy number. Concurrent analysis of SNPs at this locus revealed that SNP values could be used to predict C4 locus structure. Using this model, they then predicted C4 structural haplotypes from the SNPs of 60,000 genetic samples, half of which came from schizophrenics. The risk of schizophrenia was found to vary directly with structural haplotype, with the highest risk conferred by 2 long C4a alleles, a trait that's present in 11% of the population. This haplotype alone was associated with a 27% increased risk of developing schizophrenia. The team then explored if the brains of schizophrenic patients with this genotype would have increased C4a mRNA levels. An analysis of 35 schizophrenia patients and 70 normal individuals revealed that the median expression of C4a RNA in the schizophrenic brain was 140% greater than the normal population. It remained 130% greater even after adjusting for increased C4a copy number. Immunostaining for C4 protein in postmortem human brains showed that C4 protein tended to aggregate around neuronal synapses. Another complement protein, C3, is known to mark synapses that will be pruned during development, and in the immune system, C4 is already known to activate C3. To test for a similar relationship in the brain, the team created C4 knockout mice. C3 deposits on synapses were greatly reduced for these mice, suggesting that C4 could play a crucial role in synaptic pruning. This hyperactivity of pruning promoted by the C4 allele variants helps explain why schizophrenia onset tends to occur in adolescence, a time of extreme synaptic pruning and restructuring, as well as why schizophrenics tend to have thinner cerebral cortices than other adults. To date, most genetic findings are limited to rare alleles with large effect size. This work heralds a future in where we can better understand the complexity of phenotypes and the variable expressivity that results from the powerful cumulative effect of multiple causal alleles. But, despite this achievement, there is still much to be done. To quote Steve Hyman of the Broad Institute, "There is really light now that we can see at the end of a long, dark tunnel of ignorance. I have no doubt the road will be hard. It will be longer than we want, but I think we finally know where we're going." As we continue to improve and refine our techniques in the coming years, we may soon emerge from this tunnel of ignorance armed with the knowledge needed to treat a host of hitherto incurable diseases.